cells of opposite mating types and bring about changes in their cell surfaces so that fusion can occur. Once cells of opposite mating types have associated fusion of both the cells and their nuclei occurs, resulting in the formation of a diploid zygote. From the latter, meiosis regenerates the haploid vegetative form, and the life cycle is complete. Eukaryotic microbial diversity. We now explore the diverse groups of microbial eukaryotes. These include the protozoa, the fungi, the slime molds, and the algae. We preface the tour with a phylogenetic consideration of these organisms, where we will learn that some eukaryotes are phylogenetically highly derived, while others are apparently retained key properties of ancient eukaryotes. Phylogenetic overview of eukarya. We perceive the phylogeny of eukarya within the context of the universal phylogenetic tree. From this we learn that eukarya from their own phylogenetic domain, and as a group are more closely related to archaea than they are bacteria. This phylogeny of eukarya is deduced from the comparative sequencing of 18S ribosomal RNA obtained from cytoplasmic ribosomes. But before we examine this, let us briefly consider the classical picture of phylogenetic diversity. 5 kingdoms or 3 domains. Eukarya have traditionally been grouped into 4 kingdoms of organism, plants, animals, fungi, and the remaining microbial eukarya called protists. Before the era of molecular phylogeny, these 4 groups, along the prokaryotes, that were simply lumped into one phylogenetic unit, formed the so-called 5 kingdoms of living organisms. However, molecular sequencing has shown that the 5-kingdom system greatly magnified the evolutionary importance to metazoans, multicellular plants, and animals. Indeed, as we have already seen, the evolutionary picture that has emerged from ribosomal RNA sequencing, that the 3 domains of cellular life differs significantly from the 5-kingdom hypothesis. In particular, by showing that the bulk of evolutionary diversity on Earth resides within the microbial world, shows details of eukaryodomain of the tree of life. We can see that several microbial eukaryotes occupy deep lineages on the tree, while metazoans multicellular organisms are highly derived organisms, an observation supported by the rather late appearance of metazoans in the fossil record. Algae are scattered within the eukarya tree, mainly in relatively recent lineages, while the fungi, except for the Omycetes, from a very tight and rather recent phylogenetic unit. Of great interest of microbiologists today is the identity of the evolutionary early eukaryotes, modern-day organisms that are at least derived, evolved from their ancient ancestors. The ribosomal RNA tree clearly identifies diplomonads such as Giardia, Microsporidia, such as encephalolysosome, and trichomonads, such as trichomonads, as early eukaryotes. In what ways do these organisms resemble primate eukaryotes? Giardia, trichomonas, and encephalolysosome are all amychondriate eukaryotes. All have a membrane-enclosed nucleus but lack mitochondria. Some of these organisms, such as trichomonas, contain hydrogensomes, while others do not. Are these modern relics of ancient eukaryotes that never underwent endosymbiosis? This is one possibility, but genetic studies don't support this hypothesis. Using sensitive nucleic acid identification methods, it has been shown that amymitochondriate eukaryotes contain genes in their nuclei that originated from bacteria, just as mitochondrial genes remain today in the nucleus of aerobe eukaryotes. This suggests that the organisms such as Trichomonas once harbored endosymbionts, but for some reason discarded them later, leaving only a trace of their former existence as bacteria-specific genes in the nucleus. The Microsporidia, or evolutionary enigma, these organisms lack all organelles, including the Golgi complex and hydrogenisms, and contain very small genomes. The genome encephalidosome, for example, is only 2.9 megabases, which is 1.5 megabases smaller than that of the bacterium Escherichia coli. Induced microsporidia have all features one would predict to be present in an early eukaryote. However, because this group has apparently undergone very rapid evolution, this can be seen in the long line in the tree. Its phylogenetic position is hard to pin down using ribosomal RNA sequencing methods alone. Indeed, molecular sequencing of other genes and proteins from microsporidia tell a completely different story. Collective molecular sequencing studies strongly suggest that microsporidia are relatives of the fungi, rather highly derived eukaryotes, despite having many possible primitive properties. Further work on the phylogenetic position of this interesting group of microbial eukaryotes is in progress. The question remains, do modern relatives of eukaryote microorganisms exist today that never experienced endosymbiosis? If such organisms exist, they are predicted to lack organelles and evidence for bacteria-derived genes in their nuclei and to have branched earlier than the known early eukaryotes on the ribosomal RNA phylogenetic tree of eukaryotes. Whether such cells exist unknown, a prime challenge for scientists studying the microbial diversity of eukaryotes today. Amoeba, Paramecium, Trypanosoma, Protozoa. Protozoa are unicellular eukaryote microorganisms that lack cell walls. They are generally colorless and motile. Protozoa are distinguished from prokaryotes by their eukaryotic nature and usually greater size from algae by lacking chlorophyll, from yeast and other fungi by their motility and absence of the cell wall, and from the slime molds by their inability to form fruiting bodies. Also, a previously mentioned protozoa are phylogenetically diverse, appearing in several lineages on the eukaryote tree. Protozoa are found in a variety of freshwater and marine habitats. A large number are parasitic in other animals, including humans, and some are found growing in soil or in aerial habitats such as on the surface of trees. Most protozoa feed by ingesting particulate materials, usually other cells, by phagocytosis, a process of surrounding a food particle with a portion of their flexible cell membrane to engulf the particle and bring it into the cell. Some protozoa can literally swallow bacterial cells, or some eukaryotic cells, by operation of a special structure called the gullet. As is appropriate for organisms that catch their own food, most protozoa are motile. Indeed, their mechanisms of motility are key characteristics used to divide them into taxonomic groups. Protozoa that move by amoeboid motion are called sacrodina. Those use flagella, the mastigophora, and those using cilia, ciliophora. The apicomplexans, a fourth group, are generally non-motile and are all parasite for higher animals. Mastigophora, the flagellates. Members of the protozoa group are motile by the action of flagella. Although many flagella protozoa are free-living organisms, a number are parasite and or pathogenic for animal, including humans. The most important pathogenic mastigophora are the trypanosomes. These organisms cause a number of serious diseases. In humans and vertebrate animals, including the feared disease, African sleeping sickness, and trypanosoma, the genes infecting the humans. The protozoa are rather small, about 20 microns in length, and are thin crescent-shaped organisms. They have a single flagellum that originates in the basal body and folds back laterally across the cell, where it is enclosed by a flap surface membrane. Both the flagellum and the membrane participate in propelling the organism, making effective movement possible even in body, which is rather viscous. Trypanosoma gambinense is the species that caused the chronic and usually fatal African sleeping sickness. In humans, the parasite lives and grows primarily in the bloodstream, but in the later stages of the disease invasion of the central nervous system occurs, causing an inflammation of the brain and spinal cord that is responsible for the characteristic neurological systems of the disease. The parasite is transmitted from host to host by the testy fly, Glossina sp, a blood-sucking fly found only in certain parts of Africa. The parasite proliferates in the intestinal tract of the fly and invades the insect's salivary glands and mouthparts, from which it can be transferred to a new human host following a fly bite. We describe phototrophic flagellates in section. These are the euglenoids, flagellates that contain chloroplasts which allow the photosynthetic growth, however in darkness cells of euglena. A typical euglenoid can survive and grow as chemoorganotrophs, and such these organisms become phenotypically indistinguishable from protozoa. Many euglenoids are known, and they are exclusively aquatic, inhabiting primarily fresh waters. Unlike other flagellated protozoa, the euglenoids are non-pathogenic. Sarcodina, the amoebas. Among the sarcodinas, the organisms such as amoeba, which are always naked in the vegetative phase, and the forma infera, amoebas that secrete a shell during vegetative growth. A wide variety of naked amoebas are parasites of humans and other vertebrates, and their usual habitats is the oval cavity or the intestinal tract. They move in these habitats by amoeba movement, a mechanism also employed by cellular slime molds. Entamoeba histolytica is a good example of a parasite amoeba. In many cases, infection causes no obvious symptoms, but in some individuals it produces ulceration of the intestinal tract, which results in a diarrhea condition called amoeba dysentery amoebiasis. The organism is transmitted from person to person in the cyst from fecal contamination of water, food. We discussed this etiology and pathogenesis of